I'm going to jump straight in here, and the objective of what I want to talk about for the next few minutes is really exploring the connections that we have. We call this the nexus between marine biodiversity and climate, which are historically sort of considered as two separate things, but in fact they're very, very intricately connected, as I'll try to show you. I'm going to illustrate my talk throughout with data from Tara Oceans, which has generated a very global data set from this 4-year circumglobal expedition, begun by Eric Carcenti, who was mentioned yesterday, performed on this research sailboat called Tara, that is run by the Tara Ocean Foundation, who are here in Paris. So this expedition was aimed at exploring the microscopic world of the ocean, the planktonic ecosystems, and in addition to the scientific aspects, the foundation in particular were very interested in aspects relating to the popularization of science, education of children, and also influencing policymakers. These are issues that I don't really have time to go into today, but I'll be happy to talk about with you later if any of you are interested during the coffee break and later. So first of all, I want to talk about biodiversity. And the first problem you have with biodiversity is giving it a definition. Definitions of biodiversity, they go from A to Z. It depends if you're in a political discussion or if you're in a scientific discussion, which sort of scientists you are with. So there are all kinds of ways that we define biodiversity. In the context of the talk today, I'm not really going to talk about biodiversity in terms of species, populations, communities, but I'm going to think about biodiversity in a broader sense, which I think is useful as a framework for thinking about biodiversity in general. So biodiversity as it relates all the way up to ecosystems through to the entire biosphere, and all the way down, if you like, through to cells, proteins, and DNA, which is the underlying, of course, blueprint of what organisms are made of course. So from the definition, let's go quickly to a little bit of history, deep history. Let's think about where life on Earth that we know today, where it came from. I can be fairly quick on this because we have the beautiful theatre this morning that actually talked about the same things in a different descriptive way. So basically what this slide shows here is the origin of life on our planet beginning more than 3 billion years ago, starting with photosynthesis, and then over a very, very slow period of time, the prokaryotes, the bacteria, essentially dominated the world, bacteria and archaea. And it was only pretty later on that eukaryotic forms of life, more complex cells, were eventually began to appear. The photosynthetic eukaryotes, like green algae sorts of things, other more complex sort of single-celled eukaryotes. And then it took quite a long time where nothing appears to have happened before the first multicellular organisms appeared to appear in the fossil record. We call this sort of the boring billion, where we don't really know much that happened. But subsequent to this period, we have the Cambrian explosion about 550 million years ago, the first animals multicellular animals in the ocean, and then subsequently the appearance on land of plants, followed by animals, going up to modern times where we have the diversity of the world that we all know and appreciate, of course. Now, this has not been a slow, steady increase of complexification going from the early times up until today. But this whole route has been punctuated by some pretty major events of mass extinctions, where much of the biodiversity on the planet just disappeared. Okay, so we estimate that about 4 billion species about 4 billion species have inhabited our planet over time, and about 99% of those have gone extinct. Okay, so extinction is a natural process of the life cycle on Earth, and those extinctions are particularly strong during several of these mass extinction events, of which we appreciate 5 in particular as being particularly strong. So I'm not going to go into the details of what happened at each of these mass extinction events, but the bottom line is that they can be equated to very violent changes in the carbon cycle that are happening at the planetary scale. So that essentially involves where is the carbon between the biology on our planet and the geology. So it's when that equilibrium between the biology and the geology gets interrupted that we can see those mass extinction events. And so those can be caused, of course, by volcanic eruptions, by asteroid impacts, by release of stored methane, and so on. I won't go into the details, but that essentially we can equate these with violent changes in the carbon cycle. And one example that I want to give you is the late Devonian event, where we saw a global cooling event, reductions in CO2 in particular in the atmosphere. Reductions of CO2 is the opposite of what is happening today. So we had a global cooling event, and this is probably linked to the diversification and the proliferation of land plants. Okay, so life killed itself, if you like. So we are not the first, basically where the carbon is stored on our planet between biology and geology, and that is due to carbon dioxide being such a potent greenhouse gas. Since we're talking about the ocean, marine context, we can ask, well, do we have evidence for similar events happening between biodiversity changes and changes in the climate through marine organisms? And we do have one particular example that I want to give you today concerning a story of marine diatoms, which look like this: beautiful single-celled photosynthetic unicellular eukaryotes that contribute as much photosynthetic activity as all of the tropical rainforests combined on land. And we have an interesting event of where the diversification of these organisms perhaps led to changes in the climate. So this is today's distribution. Oops. This is the distribution of diatoms today on the planet. We see proliferation of diatoms in the ocean, in particular during the springtime, both in the north and in the south at higher latitudes. And the first thing I want to show you here is that we can use Tara Ocean's data from today's global ocean, and we can more or less recapitulate these patterns that have been observed through other studies and that are now modeled very accurately. So we can see that Tara Ocean's data recapitulates this strong high abundance in the higher latitudes at both poles. Okay, so what we can do to address where the diversity of diatoms came from using Tara Ocean's data is we can basically take all of the DNA sequences that we generate from diatoms in today's modern ocean. We can throw them onto a phylogenetic tree that has been dated, okay? And we can see where those sequences land. We can see, if they land towards the outside or if they land towards the inside. And so from here, we can generate some sort of patterns of changes of diversity over time from the beginning, from the early evolution of diatoms all the way through to the modern diatoms. And so in this study, which we did in collaboration with Hélène Morland, our colleague in EBENS in Paris, what we basically see is that the diversification of diatoms from their origins 200 million years or so ago is fairly constant, except for this pretty dramatic shift that you can see here. We have a dramatic increase in diversification at one particular time in the past, which corresponds to about 35 million years ago. So what happened 35 million years ago to perhaps provoke this? What happened in particular was, at around this time, was that the Drake Passage opened. This was the separation of the Antarctic continent from South America, which led essentially to the creation of the Drake Passage and the Southern Ocean circulation system. And we can see that this event in the geological past of the Earth correlates nicely with the Eocene-Oligocene glacial maximum, where we see a reduction in temperature at a fairly global scale happening at about the same time. So perhaps this opening of the Southern Ocean, the proliferation of diatoms in that area, which we see, they love that area, perhaps led to this global cooling, much like the plants caused the global cooling at the late Devonian period several hundred millions of years ago. So those are examples of where changes in diversity can clearly lead to changes in climate. This is evidence from the past. Evidence from today's modern ocean. Let's look a little bit about at how the world looks today. Here we're visualizing our planet from space using satellites that are capable of measuring chlorophyll in the surface ocean. They also measure chlorophyll on land, and we can see changes through the seasons on land very nicely. And we can see also changes in the ocean. We can see how dynamic that is. And from observations like this over the last 30 or more years, we appreciate now that the oceans ocean phytoplankton contribute just as much photosynthetic activity as do all the land plants, the trees on land. Okay, so the amount of carbon dioxide recycled by phytoplankton each year is very, very similar to the amount of CO2 that is recycled by land plants. So this shows us the importance of phytoplankton in the ocean, even though we don't see them because they're microscopic. So we see their importance in the carbon cycle. Another view of the phytoplankton is the opposite view, looking down the microscope. So in the microscope, we can appreciate a huge diversity of photosynthetic single-celled organisms in the ocean, going from cyanobacteria like Prochlorococcus, Synechococcus trichodesmium, through to the single-celled eukaryotes like dinoflagellates, diatoms that you've already seen, and so on. So using, again, Tara Ocean's data, I'm going to show you how we can try and put those different views of photosynthesis together. So first of all, from a genetic point of view, using the genomics data that we generate from the Tara Oceans project, and using a specific marker for all photosynthetic organisms, known as PSBO, we're able to quantify the abundances of each of these different kinds of photosynthetic organisms at the same time in the ocean on this single slide here. So here we have both the prokaryotes on the top, and we have also the eukaryotes at the bottom. And you see the abundance of each by the size of the bubbles, and you see their diversity going from yellow through to dark blue. Okay, so we can see all of that on one slide. In Tara Oceans, we also size fractionated the samples that we collected, so we can see the cyanobacteria in the small size fractions, in particular in the low latitudes, and we can see the larger organisms like diatoms, in particular in the larger size fractions, in particular in the higher latitude regions of the Arctic and the Southern Ocean. So what this becomes interesting in terms of climate and carbon, where the carbon is, because we can relate this to relative cell abundances, and we can relate that to relative biovolumes. So the biovolumes of the cells gives us an indication of how much carbon is in each of these different organisms. Okay, so since carbon is so important in the climate system, this really helps us understand where is the carbon in each of these different photosynthetic organisms. So that is useful because it can get us through to the relation between the biodiversity and the functioning of the ecosystem. This is called Biodiversity Ecosystem Functioning Research, BEF. It's an extremely active area of research in terrestrial systems, not so much studied in marine systems because it's fairly complex. But this, basically allows one to see how biodiversity per se in a given ecosystem is related to its function. So in this case, our interest is carbon fixation, photosynthesis, which ultimately has an influence on the carbon cycle and climate. So what we can do in our oceans is take the information about the different kinds of organismal groups that do photosynthesis. Each one of these is a different group of photosynthetic organisms. And we can see how each of those different groups of organisms relate to environmental parameters that relate specifically to photosynthesis. So net primary production is one that we can calculate from space, from satellite observations. Chlorophyll we can quantify based on direct measurements in the water. And what we see then is this particular group of organisms which are correlating most strongly with net primary production, with chlorophyll, as you see here, which turn out to be the diatoms, again, as we saw in the earlier studies. So this gives us an indication that the diatoms are, on a global scale, are perhaps correlating most strongly, most important for photosynthetic activity in the ocean. Okay, but we need to try to go beyond that and dig deeper to see what we can learn more about this biodiversity ecosystem functioning relationship. And to do that, we tried to couple things together, going from the gene, the PSBO gene that we use to quantify the phytoplankton, all the way up to the satellite data, of which we have 40 years of records. Okay, so I'm not going to go into the details of how we did this, but the results of this study are shown on this one slide. And essentially what we can see at a global scale, that's shown here. So we are looking here at the relationship between biodiversity on one side and primary productivity, so ecosystem function, in terms of primary productivity, how much CO2 is being taken into the system in the ocean. And so what we see at a global level is we have this sort of linear relationship, in the sense that the more biodiversity you have, the more primary production you have, the more carbon fixation you have, okay? Fine, but that only gives one side of the story, because if you break it down, if you break the ocean down, we can actually repartition the ocean into several different oceanic regions. And so, for example, the sub-Antarctic front, which is this green area here, I think, you see has a negative relationship. The more biodiversity you have, the lower is your primary production. In Antarctica, deeper into the Southern Ocean, we have a sort of unimodal relationship, where we have a sort of sweet spot of biodiversity at which photosynthetic activity is higher. On both sides it goes down. And then in equatorial regions, shown here, we have the opposite relationship, where the more biodiversity you have, the more photosynthesis you have. So this information is extremely important to tell us where the carbon is going from the atmosphere into these different organisms, and how that happens in different oceanic regions, at least in today's ocean, which I'm sure you can appreciate is pretty important information. So that is one aspect of where the carbon goes in the ocean, in terms of photosynthesis in the surface ocean process driven by light. The other very important aspect of carbon in the ocean, as part of the biological carbon pump, is the export of carbon to the deep ocean. So because the oceans are deep, these organisms, when they die or when they're consumed, when they're decomposed, a fraction of them will sink all the way down to the bottom of the ocean. So this is a very slow process, but it's an incredibly important process on a planetary scale because it's this carbon pump which essentially generates all of the oil and the gas fossil reserves that we are burning so actively today. We are probably consuming each year the equivalent of 1 million years of carbon export of plankton. Okay, so our power on the carbon cycle at global scale is like 1 million X compared to if we weren't there. So we've become a pretty important global power in terms of our influence on the carbon cycle. So again, using Tara Ocean's data, let's see what we can do to try to learn more about this process of how the carbon goes from CO2 in the atmosphere all the way down to the bottom of the ocean. And so we're basically looking to see which kinds of plankton are most strongly associated with this activity. So on the one side, we can quantify this, the amount of particulate material in the water column by quantifying it using a camera that basically goes down in the water and measures the amount of particulate material in the water column down to 500 meters. We can get global pictures of that. And we can relate that with the communities of plankton that we see at all of these hundreds of different sampling sites where we did sampling. And basically what this allows us to do is to go from these generalized ideas about plankton communities and who does what and so on, through to a more sort of hub and spoke view of how carbon export may involve different kinds of plankton, where we can identify particular hubs, particular taxa of plankton that seem to be particularly important in the process. And some of those are listed here. Some of them we expected, like radiolarians and dinoflagellates, but others were quite a surprise. Synechiophages are viruses which infect tiny little cyanobacterial cells called Synechococcus. So this implies that even the tiniest component of the system, the Synechiophages, can have a very, very profound impact on the carbon sinking to the bottom of the water column. Okay, so these are different ways in which we can interrogate which sort of organisms are involved in which sort of processes and how the biodiversity of those organisms may impact the processes that they're part of. Now I want to move forward into the future and see how we can maybe use this information from today's ocean to try to get a feel for how the ocean may change in the future. So just to put this into perspective with what I told you at the beginning of this presentation, if we put the history of our planet into a 12-hour clock, we end up with something that looks like this, with photosynthesis evolving at about 2 o'clock, all right? And then very, very little happened up until something like 10 o'clock or so, 10 p.m., where we had the Cambrian explosion. Multicellular life exploded in the ocean. And then land plants, animals appeared on land, perhaps around 11 o'clock or so. Dinosaurs appeared, disappeared, between 11:30 and perhaps a little bit later. And then finally, here we are, Homo sapiens. We appeared in this representation of the history of our planet. We appeared 2 seconds ago, okay? And if we think about the Anthropocene, which we've heard about, the Anthropocene here would represent about 1/10 of a second. So in spite of the fact that we have only been around for a very minuscule period of time, our impact on the planet is astoundingly strong, equivalent to those mass extinction events that we've seen in the past, quite possibly. So how can we address that? Again, using the example of Tara Oceans, how can we address how the future ocean is maybe going to change? I have several examples, but in the interest of time, I'll probably just talk to you about one of those. I'll skip this and talk about projected changes in plankton diversity moving through to the end of the 21st century, and the way that we can address that using data from today's ocean at the beginning of the 21st century. I'll skip that. So because we did a pretty global sampling using from the Tara Oceans expedition of all different kinds of planktonic organisms, we can expand that information out to the global ocean using modeling approaches. Again, I'll not go into the details. And we can see the general here diversity patterns of different kinds of organisms, be it photosynthetic eukaryotes or photosynthetic bacteria. And we can see largely that we have diversity being highest in the lower latitudes and tapering off as we go towards the poles, okay? And that's a typical latitudinal gradient that we see also on land over several centuries of studies. So this is sort of the diversity in the ocean, how it looks today, the beginning of the 21st century. To address how that may change in the future, we can basically take that biodiversity data from today's ocean and throw it into the ocean that is projected to occur in 100 years' time from the simulations of the IPCC in particular. So those simulations of a future ocean predict that we will have increases in sea surface temperature, not at all uniform, but some places more than others. And similarly, in terms of photosynthetic activity, which we've already talked about earlier, these IPCC models again predict these sorts of changes at a global scale. So we can take the plankton from today's ocean, throw it into this projected future ocean, and see how it repartitions out. And here we see a sort of summary of these sorts of analyses that were done by Lucy Zinger and Laurent Bopp in EBENS. And here we're basically plotting diversity anomalies, be it in positive and negative, of these different kinds of plankton groups that you saw before. So you see that there's a lot of activity, right? There's a lot of anomalies going on in these different groups at a global scale with pretty complex patterns. To simplify it, we've plotted it out here by latitudinal bands, going from the Arctic through to the equator, through to Antarctic. And we see how those biodiversity anomalies equate with some pretty important ocean parameters, let's say. Perhaps I guess we can see that better in this slide, where I'm plotting out actual maps of those three different parameters, be it carbon export in the ocean, fisheries activities, and density of marine protected areas. And basically what we see is where we predict those anomalies to be strongest for those different plankton groups. These are areas which are particularly important for carbon export, so where carbon dioxide is sequestered into the ocean and will make it down to the bottom of the ocean in the form of organic carbon, which is very important in climate regulation. We see that those bands of latitudinal anomalies are also areas where we have a lot of fishing industries. Okay, so fishing industries are perhaps going to be impacted as well by these changes. And then also in terms of the density of marine protected areas, these are areas where we try to preserve, we try to protect biodiversity in the ocean, right? These appear to be particularly vulnerable to these diversity anomalies that we project to occur in the future. So these are ways that we can use Tara Oceans data to explore in different ways this nexus between biodiversity and climate change. So to summarize, we have plankton microbial organisms appearing as particularly important in running in assuring the well-being of ocean ecosystems, assuring the well-being in general of the ocean, doing all kinds of important jobs for us. And as I mentioned yesterday, the important knowledge gaps which I think we need to address in the future are understanding better how marine ecosystems at all levels, how they respond to multiple concurrent climate stresses, be it in terms of acidification, in terms of temperature changes and other impacts, including pollution. We need to understand how marine ecosystems respond to those stresses. We need to understand how marine ecosystems interact with the climate system. I gave you the example of carbon, right? Carbon and photosynthesis being really key, with carbon being such an important carbon dioxide being such an important greenhouse gas. But there are other molecules like dimethyl sulfoxide, like isoprenoids, generated by marine organisms, which probably have very important effects on the climate system as well. And then we need to address, what is the potential effectiveness of different kinds of mitigation measures that we are thinking about in marine ecosystems. For example, marine carbon dioxide removal, how is that going to impact marine ecosystems is something that is key information that we need to address. To do this, we need new observational capacity. I think I'm running late, so we need new observational capacity. We need better models integrating information from the marine ecosystems into digital models that Martin is going to present to us shortly. And then we need more experimentation to address, for example, how mitigation procedures may impact ecosystems. We need to integrate that information, and we need more theory. And we need, through this, to advance the knowledge, the nexus between climate change and marine biodiversity, which will provide tools for better management of ocean resources, informing decision-making, and contributing to ocean governance. And let me thank the people who were really behind Tara Oceans and what I presented today. It's a big team, bottom-up effort with very little funding in the beginning, which got going because people, the scientists behind it, believed in the project, and it's been a great project to be part of. So thank you to them, thank you to our sponsors, which are shown down here, and thank you to the funders of the research that came subsequently, and thank you for your attention.